Hi, and welcome to Propel's series of interviews with innovative scientists who are seeking to understand the underlying cause of Crohn's disease. Today's interview, the third in a series, is with Dr. Mark Sundrud from the Scripps Research Institute in Miami. Dr. Sundrud is Associate Professor in the Department of Immunology and Microbiology. His work focuses on T-cell adaptation to bile acids in the small intestines. We are extremely grateful to Dr. Sundrud for taking the time to discuss with us his intriguing findings.
Thank you, Annabelle, and thank you, Mark, Dr. Sundrud, but thank you for letting us call you Mark. Thanks for making the time today to tell us about your research, which we think is really exciting and will be of great interest to a lot of people in the IBD community. And we look forward to hearing from you about it. So maybe we'll just start off with a very simple question. Maybe you can tell us about where your research, where you started, where it is now, and where you see it going in the future.
Oh gosh, okay, so I will attempt to do all that in a fairly concise way, although I've been told by a number of people that being concise is not a strength of mine. So I'm an immunologist by training. I did my PhD in an HIV immunology lab in the early 2000s. Got interested in sort of in autoimmunity and the role of T cells in autoimmunity as a postdoc at Harvard. But I really didn't have any plans or intention to, to study inflammatory bowel disease per se. I was always just interested in understanding, how these critical immune cells, these T cells, make decisions to be either, sort of tissue protective or bad guys that promote inflammatory tissue damage. And so the path that led me from sort of the pure T cell immunology perspective sort of into the intestine was really a process that spanned about 15 years. And so obviously we don't have time to talk about all of that, but I'll simply tell you that we became interested in understanding how T cells behave themselves in the intestine. When we started just asking questions about what makes a T cell in the intestine different from a T cell that's not in the intestine of that same individual. And we saw that what we initially saw was that there were enormous differences, and we were really surprised by this. So it really looked like the immune cells that are in the intestine, they're really specialized. They're really different than other immune cells throughout the rest of the body. So that made me really fascinated. And it's really we've been following the science from there. What I would say is that, a lot of our work has now become very much focused on understanding how immune responses and how T cells are controlled in either the small or the large intestine. And what we find is that there are enormous differences again. So it's not just that immune cells in the intestine are different from T cell or from other immune cells in other parts of the body, but even within the intestine, within different segments of the intestine. If they're in the small intestine, they behave and look one way. If they're in the large intestine or in the colon, they behave and look a totally different way. And that has been that's been a really seminal discovery for us, because when I went back and looked at sort of, what gastroenterologists and what the inflammatory bowel disease field thinks about, it's really that ulcerative colitis that preferentially targets the colon and Crohn's disease that is preferentially a small bowel disease are really two sides of the same coin. They're not really considered very different. And there are a lot of human genetic studies that have showed that there are a lot of genetic similarities between the genes that predispose you to risk to have either ulcerative colitis or Crohn's disease. But actually, if you look at those studies closely, those diseases, Crohn's disease and ulcerative colitis, genetically, even in some cases, have more similarity to other inflammatory diseases that don't affect the intestine at all. Okay, so both inflammatory bowel diseases look very, very similar to ankylosing spondylitis and to psoriasis and to these things. And in some cases, there's more genetic similarity between all of those diseases than there are between Crohn's and UC itself. And so, but it's been interesting because in terms of, you know, the development of new therapies for inflammatory bowel disease, you know, what you'll notice, right, is that those medicines that are used to treat inflammatory bowel disease patients, you know, they go through all these preclinical testing in animal models, and they usually get tested in clinical studies in both Crohn's and UC populations. And we don't really distinguish between the two. And in fact, a lot of times, if people become interested in you know, in mechanisms that they think may be druggable, for example, for Crohn's disease, may lead to a new therapy for Crohn's disease, they'll actually test it in an animal model of colitis. And so again, there's no current distinction made between Crohn's and UC, or even just sort of the small and large intestine. Everything is just sort of thrown together. But if you go back to your good old fashioned, you know, undergraduate anatomy, physiology textbooks, you'll find that we know that the small and large intestines are completely distinct organs, okay, both structurally and functionally. They do different things, they look different. And so we think that those distinctions are paramount for improving the way that we develop new medicines and the way that we think about and understand, you know, human inflammatory bowel diseases. And then the last point that I would just quickly make is that there's a lot of new genetic and clinical data coming out that is suggesting pretty clearly that Crohn's disease, particularly Crohn's disease that affects the small intestine, is a very much a unique genetic entity, and just responds much more poorly to existing therapies than either ulcerative colitis or even Crohn's disease that affects the colon. So again, I think location, right? This is all going to be about our interests are now really focused on developing a full understanding of sort of the pathways and all the features that regulates immunity and inflammation in the small intestine per se, because that's really where the unmet medical needs are.
And that's completely fascinating. And thank you for that. So one of the things that we read as lay people in the literature is that there is some thought that maybe there's a spectrum of IBDs. And maybe at one end you have UC, at the other end you have small bowel Crohn's, and maybe somewhere in the middle is colonic Crohn's disease or ileocolonic Crohn's disease, that it falls on that spectrum. So what is your thought about something that is kind of midway in that spectrum?
Yeah, you know, I think, you know, I think that the term, this might be a bit of a semantic thing for me, but I think, and I've heard that multiple times, right, that inflammatory bowel diseases are a spectrum. That term spectrum to me suggests that there are similar mechanisms that drive disease pathology across the entire spectrum. And certainly, you know, if you look at, for example, like biological medicines like Humira, right, like the anti-TNF medicines, right? So those work well, right, across all inflammatory bowel diseases. Again, they work less well in patients with small bowel Crohn's. But I think that generally speaking, you know, those medicines that work across the spectrum are probably things that those are just general anti-inflammatories. That's why steroids work. That's why other immunosuppressants or immunomodulators work. It's why anti-TNFs work, right? Because they're nuclear weapons that just shut down inflammatory responses in any part of the body. And of course, that comes with consequences. But again, I think that the semantics of IBD as a spectrum, while it may well be accurate, I think it probably underestimates the distinctions between the two different major forms of IBD, and particularly the disease location. Because again, I think that all of our studies are strongly suggesting that a number of pathways and molecules that we've identified and are now becoming interested in, that very potently regulates inflammatory diseases in the small intestine, really have no bearing on what's happening in the colon. And again, I think it just comes down to the basic physiological differences between the small and large intestines.
And that makes a lot of good sense, even without a deep scientific understanding. That makes sense. When we think about something like celiac disease, which is also limited to the small intestine, also intestinal, we know it's autoimmune. We know what the antigen is, right? It's really the only autoimmune disease, as far as I know, where we know what the antigen is. Do you think similarly in terms of small bowel Crohn's? In other words, do you think that there's some as yet unidentified antigen?
Not a common one in the way that celiac disease has a defined antigen, right? Like gluten. Okay, so there are other autoimmune diseases, type 1 diabetes, multiple sclerosis in particular come to mind, where it's very, very clear that the vast majority of genetic risk is contributed by some probably small set of antigens. They may not all be the same in all patients. So in type 1 diabetes, from what I've seen, I think now the current paradigm is that there's somewhere on the order of probably 10 to 20 different antigens that all target the same tissue in the pancreas, but the relative proportion or the relative contribution of each individual antigen across multiple patients might be different. But I think in inflammatory bowel diseases, there's no question that antigens are required to stimulate the inflammatory response. But very much unlike the celiac disease and these other diseases that are really driven by defined sets of antigens or an individual antigen, the data don't really support the idea that there are going to be some common class of antigens. Again, I think the general idea is that the inflammatory response is sparked by antigens derived from gut bacteria, right? The trillions of gut bacteria. And so that is absolutely required, whether or not there's an individual antigen or some small set of antigens that promote inflammatory bowel diseases. I don't think that that's the case. And the strongest evidence for that comes from genetic studies, because the genes that determine which antigens get presented to T cells usually show up as contributing significant amounts of risk in celiac disease, in multiple sclerosis, and type 1 diabetes, where there are these defined antigens. But that's the contribution of those genes to inflammatory bowel disease is very, very small. And again, it's greater in ulcerative colitis than it is even in Crohn's. And so I don't, I'm not sure that we're ever going to find, some unique set of antigens that is causative in either ulcerative colitis or Crohn's.
What you hear a lot in the patient and caregiver community is sort of this question of, are we just, by using the systemic, broad immunosuppressants, biologic medications, are we really just putting a Band-Aid? And how do we find the root cause? You hear that term a lot, root cause. Mm-hmm. And what I'm hearing you say is there may not be one across all IBD patients. Maybe this is why it's such a heterogeneous disease and presentation. But how do we think about in the future, instead of just suppressing the immune system, trying to learn more about kind of what's happening at the root of the disease for a particular individual, and finding ways to eventually address that?
I think you just answered your own question. The answer is to really investigate each individual patient as its own experiment. Okay, because again, I think, I don't know what the precise proportions would necessarily be, but for example, if you take 100 patients with ulcerative colitis and 100 patients with Crohn's disease, there are probably many distinct underlying pathophysiologic mechanisms, right? So the mechanisms that are actually that are really underlying the disease in one individual is very likely to be, if not entirely personalized, restricted to a small group of individuals that maybe have similar underlying mechanisms. But I use that analogy all the time. The same one that you said about a Band-Aid, right? So oftentimes, what I think about, or what I oftentimes say is that, giving anti-TNFs to inflammatory bowel disease patients is like it's like taking an aspirin when you get the flu, right? It's a symptom. It's not an underlying mechanism per se. It's not what's driving it. TNF overproduction by the hyperactive immune cells is a consequence of some underlying disease pathophysiology. And so our approach has really been, to try to appreciate and at least start to try to understand the heterogeneity, at least within Crohn's disease patients. Again, because we got to start somewhere, right? And so we've taken a lot of steps to try to look at, defined pathways or mechanisms that we've become interested in across, say, 200 patients with Crohn's disease, and even then subdivide those patients into individuals that either have small bowel, large bowel inflammation, or both, and then compare and contrast, the phenotypes that we see, or sort of the features that we see of their intestinal immune compartments with either healthy individuals or Crohn's disease patients. And so for one example, we identified one protein that's used by T cells in the small intestine to protect themselves from these very toxic and pro-inflammatory consequences of these liver-derived metabolites called bile acids. And this particular protein is called MDR1. It's a protein transporter that we're still trying to understand exactly what it's doing, but we know that it's very important in sort of in encouraging T cells in the small intestine specifically to behave themselves in the presence of these bile acids. And when we looked at the expression of this transporter across, a couple hundred healthy individuals, Crohn's patients with or without small bowel inflammation, or ulcerative colitis patients, what we found was that about 5 to 10% of small bowel Crohn's patients showed deficient expression of this transporter. That gives you a sense of the type of heterogeneity that we're talking about, right? So we're talking about that pathway and that mechanism may be relevant in only 5 to 10%, right? And so on one hand, that is it's maybe a less interesting mechanism from a sort of blockbuster drug type of mentality, where you want to make a medicine that's going to treat everybody, right? But I think we do have it in us in the near term, to be able to understand and improve the treatments of individual patients with existing therapies. And so that's really the goal of precision medicine, and that's really where we're headed.
Absolutely. And obviously that's something that we hope will happen. I have a little bit of personal skepticism from my pharma life, because we've been talking about personalized medicine, precision medicine for 20 years in various different, therapeutic areas. And I hope that's where we're headed. But it's also a term that gets thrown around a lot. And we hope to see really outcomes like you're talking about, is finding 5 to 10% and then, targeting that 5 to 10%, then finding another 5 to 10% and targeting that.
Right.
I think that's absolutely the correct approach. At least it makes sense to me. And I do want to head into talking about bile acids, because your work there has been really, really interesting. You touched on it. And then maybe from there move on to diet and some of the work you've done around fat, which I think is also really fascinating. So do you want to talk a little bit about bile acids, just generally what they are? And you touched on it already, but how your research relates to that and what you found, including with your recent paper that you just talked about.
Yes, absolutely. But before I do that, I wanted to come back to the point that you made about your healthy skepticism, right? About this notion of personalized medicine. As somebody who has been looking for ways to fund investigational clinical studies with safe, inexpensive, generic, FDA-approved medicines in small bowel Crohn's patients, and using that to understand the personalized responses of Crohn's patients to those medicines, and trying to get that work funded through traditional mechanisms, it's been really challenging. But this is where it's really important, the work that you guys are doing and the work of your supporters of this foundation. You guys are the ones that are going to change how this operates, because you're absolutely right in, there's a much lower return on investment for a medicine that can only be used in a tiny subset of individuals. But imagine the impact that you can have on individual patients by giving them safe, inexpensive medicines that really effectively and durably treat their disease, right? So to me, that's way more important than what comes out of it in terms of the royalties and all this other stuff, right? It's got to be patient focused. So I think the work that you guys are doing and foundations like yours are going to really help change that, hopefully. And that's really going to be a collaboration between the scientists that are interested in personalized and precision medicine and these foundations. Okay, so bile acids. Yeah, just to switch gears quickly. So bile acids are really fascinating. They're a very, very fundamental class. They're a large group, large constellation of metabolites that are made by hepatocytes in the liver breaking down cholesterol. So they break down cholesterol into these bile acids, and then they transport them out of the liver, and then they stick in your gallbladder, where they get stored there until you eat a Big Mac or some meal, right? And then when you eat, then there are hormones that are secreted that tell your gallbladder to squeeze, and that contracts and results in the deposition of these bile acids into the upper portion of your small intestine. And so the entire purpose or a major purpose of bile acids is to then tear apart all the dietary fats, all the lipids, and all these things that are insoluble. Okay, they have to tear them apart in a process that's called emulsification. So they emulsify these fats and then bring them into contact with the epithelial cells in the small intestine to allow them to absorb these dietary fats, and also fat-soluble vitamins, right? So vitamin A, D, E, K, all of these fat-soluble vitamins that we all need. You have to have a process to make these fats get solubilized and get absorbed. And so that's really the fundamental purpose of bile acids. Okay, so any lower organism that has a gut has bile acids, right? You have to be able to do this. But there's really been, so I would say that that's sort of all we thought about bile acids for, decades. And it was just that there are these boring things that help you digest fats and absorb fat-soluble vitamins. But then there was really a renaissance in people's interest and scientific interest around bile acids, starting probably in the 1990s. when a number of different groups identified that bile acids also had very important hormone-like signaling properties, which they achieve through binding to a bunch of genetically encoded cell surface and nuclear receptors. So there are these receptors that cells express that have evolved to essentially sense and respond to the presence of bile acids. And so that's now become very important because it's been shown that a number of cell types, both immune cells and epithelial cells, actually have these bile acid receptors, and that the presence, the relative presence or absence of bile acids at different times of day or whatever, all change the way that these immune and epithelial cells behave. So that's a little bit about like very fundamental aspects about what bile acids are. I think a lot of us are used to thinking now, right? We're used to thinking about all these trillions of gut bacteria, right? That we all call the microbiota, right? And you think about how dynamic those are, how different they are from person to person. Think about bile acids exactly the same way, okay? Instead of being trillions of different bile acid species, there's about 80 to 100 different bile acid species. But for sure, if you looked at, if we did a profile of your intestinal bile acids, right, of all four of us, they would all be different. Okay, so they're very much personalized. They also, bile acids have sort of these two-dimensional interactions with bacteria. So they also have bacteriostatic properties, which is why you have a lot less bacteria in your small intestine than your colon, because that's primarily where bile acids are found, is in the small intestine, and then those get actively reabsorbed and sent back to the liver in a process called enterohepatic circulation.
So that's great, thank you. And I'm sure I'll have to watch that back a few times to fully grasp it all, but it's a really interesting area. And again, it's one other piece of the puzzle in Crohn's disease. So how do you, one of the things that, again, patients and caregivers think about a lot is diet, right? We think about, well, this is a digestive disease. It stands to reason to many of us that there is some dietary component, and maybe we can even, potentially improve our health, whether it's IBD related or overall with what we eat. So what's your thinking around diet, around the Western diet, around fats in diet, and how they may or may not be related to Crohn's disease?
Yeah, no, I mean, I think, there's no question that diet is going to play a critical role in determining, either risk in inflammatory bowel disease or even disease course. Again, I think just like the question about antigens and about gut bacteria, the specifics is where it becomes a real challenge. Generally speaking, again, since we think a lot about bile acids, and I mentioned that that bile acids get deposited into your intestine when you eat, specifically in response to when you eat. It's also true that the cells in your liver make more or less or even different kinds of bile acids in response to the type of food that you take in, okay? And it's also true that gut bacteria can also act on the bile acids that your liver makes, and they can change the chemical makeup and the functional activities of these bile acids. And so if you have different bacteria, you have different food, and you have different bile acids, all three of those features are very tightly linked, and they're very responsive to diet, okay? I think where we need to be able to move in the field is getting into more specifics, right? Which bacteria, utilize, say which dietary fibers, for example, and how does that change individual species of bile acids, and how does all of that then in turn impact the function of these cells in the intestine that are driving inflammation or promoting tolerance, right, or healing? Those are the real questions.
Yeah, so is it your thought that maybe in the future, whether it's by dietary manipulation or microbiome manipulation in a therapeutic way or fecal transplant type way, that we may be able to impact kind of this cycle or this environment of bile acids and the microbiome and the immune system, and how kind of they're all dealing or how they're all working together in the environment of whatever location you're looking at.
Yeah, I mean, for sure. I think that I think there's already pretty strong evidence coming out of clinical studies that are looking at different dietary interventions. And some of those studies have looked really promising in terms of manipulating diets, especially in patients with small bowel Crohn's disease. What I would suggest is probably something that's maybe a little bit provocative, but what I would suggest is that these dietary interventions are going to have preferential effects on patients with small bowel Crohn's disease, because that is what the small intestine does, right? Is it digests food and it drives nutrient absorption. That is not what the colon does. Okay, so think about the way that we think about this. If we're trying to simplify this a little bit, we think that the bacteria-centric therapies, whether you're talking about fecal microbiome transplantation, right, or actually manipulating some specific species of enteric bacteria, we think those will probably have very important therapeutic implications in ulcerative colitis and large bowel Crohn's disease. But we really think that this interplay between bile acids and the diet is going to be paramount in controlling small intestinal inflammation.
That's great. I know we only have a few minutes left, so I just want to open it up to see if Natalie or Annabelle have any questions as well.
Yeah, I'd like to ask, do you believe that there's a possibility of some sort of pathogen that is one of the triggers of Crohn's?
I mean, certainly it's not out of the question. I don't necessarily tend to think that, again, that we're going to find one common individual environmental trigger for all individuals with all forms of IBD. But some sort of trigger, obviously, I think is going to some sort of environmental trigger, whether it be an infectious trigger like a pathogen or a dietary trigger, right? Something like that is for sure going to probably be found to be a very important underlying factor in sort of taking individuals who have genetic predispositions and then drive those people into full-blown symptomatic disease. And one thing that I would point out is that there are a lot of really great studies going on. In particular, I'm aware of a couple from groups up in New York that have put together these really amazing patient cohorts and are doing all sorts of exciting studies where they're taking, say, unaffected first-degree relatives of patients with inflammatory bowel disease who are at exponentially higher risk of developing inflammatory bowel disease, and actually studying them both before and in some cases after the onset of overt inflammatory bowel disease. I think that's where those sorts of questions are going to become, are hopefully going to be addressed, and I think are super exciting.
Yes, and the GEM study in Toronto as well,
Yep.
where Natalie is.
Absolutely, absolutely. Our collaborators at the University of Miami are involved with that as well.
It's a great study, and hopefully it will answer many of these remaining lingering questions.
Well, thank you. I know, again, we're short on time, so I just wanted to thank you so much for this absolutely fascinating discussion. I think we could all listen to you talk all day. And we completely agree that there's a need for funding for novel, innovative research like yours that isn't necessarily going to be the most profitable research, but is going to help patients, and that's our goal. So we want to thank you for doing that work, and want to thank you for this excellent, introduction or insight into your thinking. And with that, maybe we'll just end it here.
Yeah, absolutely. Thank you guys again so much. It's been an honor talking with you guys, and keep up the great work.
Thank
Thank
you.
you. Thank you so much, Mark.